Project Catalyst presents Bench to Bedside Chats. Direct from Building 22, White Oak Campus, FDA. Brought to you by the Oncology Center of Excellence under the direction of Dr. Richard Pazner. Beyond these doors, the Oncology Center of Excellence, in collaboration with the Centers for Drug Evaluation, Biologic Evaluation, and Devices and Radiologic Health, and most importantly, dedicated people to include scientists, regulatory reviewers, and leaders, have developed multiple programs focused on improving the treatment and cure of cancer. Project Catalyst is one of those efforts intended to help the academic scientist, university life science accelerator, and oncology startup company navigate the regulatory drug development enterprise to translate your insights into drugs to treat and cure cancer. So I am really excited to have Dr. Booth and Dr. Zhang here today for Project Catalyst. And today we're going to talk about pharmacology in early human, first-in-human studies. And, you know, I've been at the FDA for a long time. I've known Brian probably for 20 plus years here at the FDA. And I've only got to recently meet Dr. Zhang here in the last few days. But I think that they're going to be able to provide us some really useful insight on early pharmacology studies. And I just wanted to kind of set the stage a little bit for kind of what these bench to bedside chats are intended to do. Our audience is, you know, early startup oncology companies, academicians who are thinking about trying to develop an anti-cancer therapy, whether that be a small molecule drug or a well-characterized therapeutic biologic protein. We're going to try to use these bench to bedside chats to help provide some clarity to various guidance documents. I mean, one of the things I say many times when I give talks to the external stakeholder community is you really have to read those guidance documents, and FDA puts a lot of time into those. But sometimes throughout the process of editing and clearing, it can become somewhat abstract and maybe even, you know, obtuse sometimes. And so hopefully this, you know, for someone who's not a aficionado and hasn't been around the block 100 times, sometimes we can provide some granularity to those guidance documents. And we also wanted to be able to have a kind of an oncologic perspective, because oncology is really different with regards to most of the other drugs in OND, because they're truly life-threatening, imminently life-threatening conditions. So we wanted to kind of put that risk-benefit, you know, assessment around, you know, our thoughts. But one of the most important things here is, you know, even though you may be a small company, the same regulatory standards apply to you as a small company as to you know, a large pharmaceutical company. There's not a different, there aren't different laws, there aren't different regulations for you. And with that, I am going to I also want to make it perfectly clear, we're not here to develop new policy. We're not here to, you know, make new regulations or to, and most importantly, we're not here to set the bar to the point where people see that, oh, here's the threshold that we need to jump over. The idea is to really help get concepts across and how the agency thinks about various, you know, aspects of drug development, so that you can approach it in a more, you know, informed and enlightened fashion. So with that, I'm gonna let Brian introduce himself, and then let him introduce Dr. Zhang, and you guys can tell me a little bit about yourself, how long you've been here, and what you guys do.
All right. Well, good morning. Thanks for having us, Jeff. So as you said, we've known each other for some 20 odd years or so. I'm currently in my 26th year here at the FDA. I'm the director of the Division of Cancer Pharmacology 1 in the Office of Clinical Pharmacology, and we support oncology drug development. I've been in oncology ever since I joined the agency. And I have with us this morning Dr. Nan Zhang, who is the team leader in our group. And She's got a background in pharmacometrics and came over and joined us in oncology a couple of years ago. And Nan, perhaps you want to say hello.
Hi, this is Nanjun. I'm a team lead working with Brian in the Division of Cancer Pharmacology one.
And where were you before you came to the FDA?
I came directly from graduate school, but I worked in the Office of Generic Drugs for about 5 years before joining the Office of Clinical Pharmacology.
Awesome.
And within the Office of Clinical Pharmacology, I worked as a pharmacometrics reviewer and also the QT lead in the office for a couple of years before joining the division. While I was working with pharmacometrics and the QT group, we do see a lot of special, unique features for the oncology drug development, and that interests me to explore further in this area.
Yeah, exactly. Hopefully, I mean, and as I was going through this, I I always thought of pharmacology as a much later part of drug development, and I never really, because I always thought it as the absorption, distribution, metabolism, excretion, and that part of that first in human trial, it seemed to me, and to the night, for someone who doesn't do this on a regular day-to-day basis, and just sits in meetings and rubs their chin and nods like they have some understanding of what's going on, that I didn't appreciate how much pharmacology has to play in really truly in first in human trials, and whether or not you conduct a successful first in human trial with regards to both biomarkers and pharmacodynamics. And I assume kind of the impetus for this also was over the last number of years now, we've really tried to have a kind of new focus or perspective on optimal dosing, right? And trying to shift the paradigm of, oncology therapeutic development from that maximally tolerated dose, right? So how, did pharmacodynamics really matter that much? We were going to dose till we got toxicity, and then we were going to move that forward. Whereas now there's a lot more insight and thought in, are we hitting the target? Is a drug getting in the cell? And I think you guys can have a lot to say on that for this whole dose, Project Optimus and dose optimization as to how you would think about that early in first in human trials. But I guess what I would first question I probably want to ask, because there are a whole bunch of different new guidance documents that have come out in the last 3 or 4 years or so. But if you two had to say, for pharmacology, what are the, for early stage companies, small pharma, small companies, academicians, what are those key aspects that they really need to think about to have a successful first in human trial from a pharmacology perspective? You guys, if you had to, like, if there was one concept or a few concepts to have people really think about, what would those be?
Sure. So before I move into that, I want to come back to your guidance remark. As you've noted, we have a number of different guidances, and from my perspective, there's sort of broadly two types of those that we have in clinical pharmacology. Okay, so we have some that deal very specifically with how to do certain aspects. So like how to conduct a mass balance study, how to do drug-drug interaction studies. So these are guidances that look at doing specific studies to answer specific questions. Okay. And these are sort of like the base questions that we have in clinical pharmacology. So kind of the second set of guidances we have are sort of more topical. So we have guidances on, say, ADCs, antibody drug conjugates, and we're asking those basic clinical pharmacology questions for this special class of drugs and how to address this for this particular class. So you have these two broad types of guidances that we use in the Office of Clinical Pharmacology. I wonder if I might show a
absolutely
picture that we made, because I think it's helpful in describing what it is
Before you start there, can I just throw in the comment of that truly, I mean, if if you're going to develop a drug, and it doesn't matter where you are in small pharma, small company, startup, academician, you really need to step up to the plate. And when people haven't read the guidance documents, I mean, that's frustrating for me. So that's really like the first place to kind of start, right? Is to read those guidance documents.
It is. And actually, when you get into interactions with the FDA on your particular application, we get involved with investigational new drug applications. And generally in our responses to those kinds of inquiries, we also append at the end a list of applicable guidances that may be relevant to the development of your product. Okay. And I don't know if Nan wanted to add a few words on that. But that's something that we do regularly, and we do it to try to be helpful to the people we're interacting with, so they can get a little more focused on the things that are relevant with respect to the clinical pharmacology development of their products. Nan, I didn't know if you wanted to.
Can I jump in there, Nan, before you say something? Because part of the reason I wanted to have this particular bench to bedside chat was I was always overwhelmed with, I mean, like you said, there's the ICH E14 QT. So I hope that, what you guys could do is provide me a list of the ones that you have to do to get to the first in human trial or to get through your first in human trial to be successful. Because
I think that, Nan, did you want to take a stab at some of the guidances that should be focused on around the first in human studies?
I think first of all, I think depending on the drug molecule that is under development, there could be a different set of guidances that may be more relevant to this specific sponsor as compared to the others. So first thing would probably be still understand this molecule from the non-clinical side and from the chemistry part. Other than that, I think most of our guidances, selected guidances that is attached after the initial interaction is usually generally applicable. To prepare for the first in human study, I think the general concept about patient selection that may involve some thinking on the potential drug-drug interaction and the patient selection organ impairment, those will always apply, regardless of the molecular type that will be chosen. Some other ones, like bioanalytical methods, development, food effect, those may be kicked in at a later time point, but those concepts I think should be kept in mind throughout the development.
Okay, and is there something a distinction to be made between whether you're looking at a small molecule drug or a large molecule drug like an antibody type of drug? Are there different things that should be considered with respect to the guidances when you're getting started out in these early phase trials?
Small molecules and large molecules, in terms of drug-drug interaction potential and potential PK impact by organ impairment, we do, based on prior experiences from those classes of drugs, we do have some different expectations. So those are the things I think sponsor can pay attention to when they come to the first in human study protocol development. And also with regard to the QT risks and the dosing instructions, those could be subtle differences in the study design.
So let's turn to the figure I put up here for a moment, because this is like a very high level summary of what we're trying to accomplish in clinical pharmacology. And I think it's probably helpful to take a look at this. And so really, our main thrust is really about identifying the dose that's going to be used in the patient population. And currently, as Jeff mentioned a little bit earlier, we have a lot of emphasis in oncology of trying to find the optimal dose. That's the dose where we've maximized the benefit at the point where we also have the least amount of toxicity. Okay. In clinical pharmacology, we've been always about trying to find this dose for patients, regardless of the therapeutic area. But in recent times, we've really refocused our efforts in oncology, and I think that's starting to pay some dividends. Now, around this dose for the patient population, there are some general things that can happen in patient treatment that can have an impact on these, okay? And so the first of these are food effects. If you have an oral medication and you have to make a decision about whether you give this with food or not to the patients, food can do one of three things, okay? One, it can increase the exposure of the drug concentrations. That can lead to increased risks of adverse events. And so that's something we have to be careful of. Sometimes food for some drugs can also reduce the absorption of the drug, and that can lead to a loss of efficacy. So that's another condition that we want to be wary of. And then sometimes, and this is the condition we hope for the most, food has no effect on the drug. And so you can give the drug without respect to taking it with meals or not. And that's, the ideal setting. That's the thing we strive for. So these other three bubbles show some clinical conditions that can arise in patients. They're very common, right? So oncology patients are often on a number of different medications. And so these can interact with the oncology drug. And you can have situations where, again, you're increasing the drug concentrations of the oncology drug, which puts you at risk for adverse events. Sometimes we have some drugs that can induce the metabolism, and that can lead to reduced concentrations. So you have the risk of a loss of efficacy. So we need to study these kinds of interactions in the development of the product. Furthermore, for small molecule drugs, they're often excreted either by the renal or the hepatic route, sometimes both. And if you have organ dysfunction here, what can happen is you can end up with high levels of concentrations again, which predispose you to adverse events. So again, here we may need to look at clinical pharmacology, and we may end up with some kind of a dose modification for these three kinds of conditions. Now, one of the major tools that we use, there we go. One of the major tools that we use for assessing these kind of scenarios is exposure response, right? So we try to plot drug dose or drug exposure against its effect, whether that's efficacy or safety, okay? So we develop these relationships, and you can see efficacy is shown here in blue, and toxicity is shown here on red. And so by understanding these fundamental relationships for each drug, we can then rationally go and try to pick the optimized dose. So for instance, in this case, here we have efficacy, which is just about as high as it's going to get. And it's at a point where the toxicity is lowest, okay? And that's better than selecting a dose that's higher, where you might have a little bit more efficacy, but you can see that the toxicity rates are much higher. So that's less desirable, right? So this is the calculus that we're trying to make in each of the drugs that we look at. Now, if we move to the next slide. So this is an overview of drug development in oncology. I mean, you could apply this really to any drug in any therapeutic area, right? So we start out in the discovery phase where the drug company characterizes some molecules they want to bring forward to treat a particular disease. Then we start clinical trials, right? And this is our first in human study. So we move through phase 1, at which point we get an idea of the kinetics and the tolerability, and in many cases, a little bit about the activity or the efficacy before moving into phase 2, where we get more information on the dose that gets selected. And then typically, or often, we go to phase 3, right, which is the main trial to the overall safety and efficacy in terms of patient outcomes. In oncology, we often will follow this accelerated approval pathway, right? So we approve the drug based on a biomarker that's reasonably likely to predict clinical outcome, and we submit an NDA early. And as a part of this pathway, there is a confirmatory phase 3 trial that gets conducted later to show the clinical outcome of the product. Now, when you take this pathway, this accelerated approval pathway, one of the things is you can see all the things you need to try to find out about the drug are compressed into a smaller time frame, right? So right away, there's a very high onus on trying to figure out the right dose very early in development, right? Beginning right at this first in human study. So this is something that we typically see in the phase 1. It's a dose escalation trial. You have different doses that get tried in different cohorts of patients until you get to the point where you think you've identified a dose or doses that you want to take forward for further development. And in this case, they often go into an expansion phase, right? So they'll take that dose and they'll add more patients to it to get a better assessment of the efficacy and the adverse events. Sometimes when there's some uncertainty at this point, you might even see them take 2 doses forward to make those kinds of evaluations before fully developing all the clinical data that's needed for this accelerated approval. Now, in terms of those other clinical pharmacology things that I mentioned in the previous graphic, we have seen people who have been very innovative, right? So most of the time, people are developing these drugs in the fasted state, right? But some people have done their trials and they've included a cohort in which they've also had patients who were fed, so you can get an assessment of the effect of food on the kinetics and the behavior of the drug, so you can help make decisions earlier on about, whether you're going to need to give this patients in the fed state or faster, or whether it doesn't make any difference. Another thing that we see is that sometimes you'll also see a cohort with a drug-drug interaction portion to it. Okay, most commonly we probably see this in cases where you're developing drugs for something like glioblastoma multiforme, where many of the anticonvulsant drugs that the patients take can induce the metabolism of the drug, or in AML, where many of the patients are being treated with antifungal agents, which can inhibit some of these enzymes. And so these can change the exposure of the drug, right? And so getting an early handle on this helps developers figure out, how they want to dose, if they have the condition, how they want to dose around that or adjust to it, okay?
Can I interrupt there? One of my questions here would be, this is obviously, it's a compressed schedule. There's lots to do. It would be great if they had all the resources in the world to do the food effect study up front, to do the drug-drug interaction study up front. But if I wanted to get to my first in human trial and my value inflection point is some kind of activity, whether that be a response, or I hit my biomarker, in my assay, would those be necessary, or we're just trying to paint the most optimal picture?
Right. So, what I'm telling you about here are, some of the more creative approaches that we've seen in recent times, but they're not always done this way. Okay. So, a lot of the times people will do the dose escalation phase as you described, because they're kind of in a rush to get to that, answer that ultimate question, right? What's the dose to be using in patients? And, so what happens is they'll often do food effect studies and DDI studies in parallel to these phase 1 studies. And when I say in parallel, and the timing is that's really more up to the developers to make those choices. So it's sort of in this range between the phase 1 and 2. You're also getting this information sort of as a parallel track. We see that a lot. Okay, and that's acceptable. For the renal and hepatic impairment studies, okay, we've traditionally seen standalone dedicated studies, and these tended to be done later in development. Okay, and a good decade ago or so, these weren't done until the post-approval setting, which isn't isn't great. It's not something we advocate, but it was done that way. Now, and we tend to see these done earlier, but when we're following this shortened pathway, these generally aren't complete by the time we see them in the application, but we have some information from them, okay? And another thing that we see is that in these phase 2 studies, they've changed the inclusion and exclusion criteria to allow patients with different degrees of impairment. So it's actually quite common to get studies that have some renally impaired patients and hepatically impaired patients, usually in the mild to moderate range of impairment. And that combined with these dedicated studies gives us some information to work with about how to deal with these kinds of situations. So that by the time people get to their NDA BLA in this period, following this sort of pathway, we have information about the dose that's been selected, which we hope at this point is optimized for the indicated patient population. We have information about food, and we know what the implications are for the clinical pharmacology and how we're going to dose the drug to patients with or without respect to food. And we have information about drug-drug interactions and renal and hepatic impairment, such that we can make some labeling instructions for physicians to understand whether they need to avoid these kinds of interactions or whether there are dose modifications for these kind of conditions for patients who have these kinds of issues. So that's just a very high overview of what we generally see in terms of clinical pharmacology development. And as you mentioned, there are a number of other guidances. Some of these support these different particular questions that get asked.
For folks who want to have a successful IND submission, right, and not get put on hold, are there actual reasons, with the idea that we're thinking more along the idea of dose optimization, more along the role of biomarkers, is our drug hitting the target? What would be pharmacological reasons that someone would not be able to move into a first-in-human trial, like that radiologic,
the mass balance studies?
Mass balance. I mean,
right.
Do we even do those in oncology? That shows my naivete here. But maybe I just never knew to look for those.
Okay, so with respect to the mass balance studies, okay, this tells us what routes of excretion are important for the drug, right? And it tells us some information about what gets absorbed and what metabolites you generate, okay? And that would support, the decision about whether you need to do a renal impairment study or a hepatic impairment study.
Absolutely.
Or both. And again, those aren't critical to starting your first-in-human study. Okay, but they are important because they underscore the decisions that you're going to need to make about what to do with these. Do you need to do both? Do you need to do one? So it's sort of helpful. And we have a number of drugs like guidances that deal with issues like that. So we have the guidance that deals with in vitro metabolism of the drugs. Okay, and that tells us about whether or not we need to look for drug-drug interactions in patients, right? And these are in vitro studies. Those are often done before you're starting your phase 1 study. Most large companies have organized their development paradigm such that they do those early, and they know right ahead of time, longer term, what sort of studies they're going to need to do to address things like P450 interactions or PPIs. Things like that.
A lot of this, like mass balance studies, are done in healthy volunteers. It seems to me like lots of this stuff is done in oncology patients, right? Because of the nature of the drugs.
Yes.
Or
yeah.
Man, do you want to?
Yeah, I think this may be a feature in the oncology drug development as well, based on the molecules properties, the toxicity profile that we can expect from non-clinical studies and from experiences with similar drugs in the area. Some of them cannot be conducted in healthy volunteers. I think this will be one of the major reasons why many of the first-in-human studies are starting in the patient population. And because it is conducted in the patient population, we do want to have reasonable estimate on the expected efficacious range so that we do not expose patient too much to an inactive drug and the patient can at least get some benefit. And I think this could be like one of the major differences from most of the first-in-human studies for oncology drugs as compared to the first-in-human studies in the other therapeutic areas. Also, while Brian showed a very comprehensive plot about what first-in-human studies can be utilized to explore many of the clean farm properties, I think some of the components are not necessarily needs to be conducted when you first submit the first-in-human study protocol. Many of the aspects like the food effect cohort or DDI cohort, they can be added via protocol amendment as we gain more information about the drug's clinical pharmacology properties, because we do need those informations. For example, the drug exposure, Tmax, half-life, those information to help design those preliminary DDI or food effect study cohorts down the road. So don't be intimidated that you need to get everything ready.
Yeah, well, I'm going to try to highlight, go back to that, because I know I mean, we think about the end game, right? That's our job, right? We want to approve safe and effective drugs, and we want to get everybody to the end game. And everybody's not going to get to the end game because every drug doesn't work. Right. You know, and I am of the opinion that, you know, today's targeted drug development, that, you know, we should have less unsuccessful trials, but there's still going to be unsuccessful first-in-human trials. Mm-hmm. But one of the things one of the things I think that would be interesting to discuss is you don't want to throw the baby out with the bathwater. You know, you want to know that your drug didn't work, that the pathway was the wrong pathway, that, you know, either you weren't hitting it despite whatever drug level you were able to achieve without toxicity, or you were hitting it and that pathway didn't stand up into a clinical trial to be effective. So it seems to me part of doing intelligent drug development, even for a small company, is that you want to do the ADME stuff, right? Because you want to know what the concentration of drug was in the patient, in the serum, in the plasma, but you really want to know what it was inside the cell too. Have you ever seen, I'm just curious, have you ever seen anyone in a first-in-human trial actually do some kind of mass, you know, biopsies and mass spec analyses of drug concentration intracellularly? Or is it that you have a particular biomarker and that you can do that biopsy and look at the effects on the biomarker? Because those are key questions for one of the pillars of good drug development.
Yeah. Unfortunately, measuring the in situ concentrations, the cellular concentrations of drugs, is currently impossible to do. Okay. So we use plasma concentrations as a surrogate for what's happening in the cells. Okay. And we'll often correlate that with other biomarkers. So for instance, one of the things people will often do is they will, even in the preclinical setting, they'll do studies to show target engagement, right? What the IC50s are, what the receptor occupancies are. And sometimes in clinical studies, they'll go and they'll get that same kind of information, right? And they'll tell you at a certain dose, they're achieving 90% target occupancy. Which is a biomarker that tells us, you know, you're pretty close to having your maximal effect, or it may imply that, okay, as opposed to data which shows you've only got 25% target occupancy at a certain dose, and so that would tend to make you think, you know what, we probably need to go higher if we're going to make this thing work, right? And you know, this can get this is a very complicated part of the calculus that goes into it. We don't just rely on that, right? Because then we also look at the clinical data. We're looking at the ORR generally, and we're looking at adverse events. And in some situations, you can get to a point where you have 90% or 95% target occupancy, but you're only seeing toxicity, so then you have to ask yourself some questions. Can we keep going with this? So this is all part of the thinking that goes into assessing, whether you get to the point where you've got the best dose available.
Add something relates to Jeff's question about biopsy and things like that.
I just wanted to please. I want to hear your thought, but I'm going to go back to Brian, and because I give these talks at different institutes, and I realize that I haven't seen it. You guys haven't seen it. But I thought that there were some mass spec ways to look for maybe they're available in the lab or even in PDX models or something. But the idea of using them in a human clinical trial to look for drug engagement of target and actual in situ drug concentration isn't feasible. Is that the?
You are correct that there are some methodologies that are, I would say, under development that look like they can get single molecule determinations. Okay, but that so far is pretty far from prime time. I don't think we have seen any application of that in anything to date. But we've had some inquiries about it. But I think we're still need that to develop
with the immuno-oncology paradigm changing PD-1s and other manipulations, modifications of the immune system. I mean, we do a lot more biopsies and to look for various biological effects. Are we bringing in CD8 T cells or NK cells? Is there a cytolytic test? And so it's almost like almost expected that you would want to develop that science. Do you ever see a point where we would expect folks to actually be doing much more pharmacodynamic target engagement kind of biopsy studies?
So I think there's a distinction to make about whether you're dealing with a solid tumor type of oncology versus a hematological one. So in the hematological setting, those types of biopsies are far easier to access than a solid tumor one is, right? So we can take serial blood samples, which is what we need to do to look at the pharmacodynamics or the pharmacokinetics. Okay, but that's much harder to do with a solid tumor. You can't take serial biopsies from these patients. Okay, so that part of the world is still hard. But in the hematological oncology part of the world, it's much easier, and so we do make better use of we do take advantage of the fact that we can do these biopsies in a more serial way, right? And in the solid tumor setting, there are efforts to try and identify other biomarkers that are related to the disease that might be soluble in the blood or the plasma that we can access better. So we try to do those things. Some of the ones that we have that are I guess older, like PSA and prostate cancer. And other things of those natures, and people are doing research in particular diseases to find better biomarkers for those particular diseases, and probably in the shorter term, those are going to offer better options than sort of in situ measurements. I think that's still a little ways out.
A little science fiction.
No, not science fiction. But it's still early in its development.
The serial biopsy stuff I understand, but occasionally allowing for a biopsy where you had you were at Cmax or you had you were on the curve and you had long enough drug exposure that a biopsy to look for target engagement and in situ concentration of drug doesn't seem completely it seems like that would be depending on, if it was a readily accessible tumor, there weren't reasons that you wouldn't want to do a biopsy, that might add value, particularly to, have we reached the right dose. But you guys, those will hopefully be being done preclinically in some kind of animal models that may translate into the human model, but we don't necessarily test those in humans.
We do see applications with a proposal to conduct biopsy and collect data from tissue samples. But I think whether it is appropriate should be a case-by-case evaluation, depending on how complicated the procedure is, how invasive it is, how much harm it may be done to the patient, especially in the very early stage, first in human studies. And also another factor we want to consider is even if you collect this data, how consistent those can be collected throughout the development program and to be further validated and to support dose selection in the long run. I think those needs to be evaluated based on individual application.
Yeah, I can't disagree. I think the more we're very data centric, obviously, at the FDA, and we like to have the data. Sometimes you just need to have, if you're making a go, no go decision, you're spending, $500,000 a patient. Sometimes even a little bit of information, it may not be enough to convince a regulatory agency, but you might, if you were at your Cmax or your AUC was where you wanted it to be, and you had a you had a protein degrader drug that was a funky, unwieldy molecule. Knowing whether or not it was getting in the cell at all might be important to your drug development decisions. Whereas I realize that we may not think that the totality of the data is very persuasive because there's so little, but it might inform the sponsor to some extent.
Can we go back to your point about the studies being done in patients? Because I think one of the things to point out, Nan was talking about some of the issues around oncology drugs and why we often do these clinical pharmacology studies in patients rather than healthy volunteers. And it's because of the toxic nature of many of the molecules, so if they're mutagenic or teratogenic, we don't want to expose healthy volunteers to that. Or if it otherwise has some toxicity that's, quite difficult to deal with, you don't want to deal with that in healthy volunteers. But, a lot of the times, the toxicity profile of drug is not that daunting. So we do see some clinical pharmacology studies that are done in healthy volunteers. You'll often see like the drug-drug interaction studies done in, well, maybe that's not a good choice, but you could see mass balance and other things done in volunteers rather than in patients. And again, those would be done in parallel to the drug development paradigm. And then you would bridge that information from the volunteers to the patients. Okay, so oncology drugs are not exclusively developed in patients. People should understand that given the right toxicity profile for the drug, it may be acceptable to do some of these studies in healthy volunteers.
And I hate to perseverate on this, but the target audience are, small companies, academicians who are trying to, determine whether or not their drug is going to be useful to continue to pursue and develop. Mm-hmm. For specific, IND enabling pharmacology thoughts, the things that they need to have in place. Like one of the things that has come up in the past for me that was very confusing was, so let's say you have a nanoparticle or it's a liposome and it's a targeted liposome with a component that's integrated into the membrane, and then there's a MER RNA inside the liposome nanoparticle, and then there's this comment that's test-free drug. or monitor free drug. And I'm like, well, what is the free drug? Is it the MRR? Is it the target?
Is you have to identify what the active moiety of the drug is. That's really what you want to measure in so much as you can. Okay, so so from the outset, we tell people, once you're getting into clinical studies, you need to have an assay that's validated. And what that means is that you've proved that the assay can measure the drug reliably. And so the data you're getting is reliable. Okay, it's believable. Okay, we don't have to worry about that. But the crux of the matter is, what exactly do you measure, right? And so in these complex, so a small molecule drug is generally pretty straightforward. You just measure the drug. And sometimes if it's a prodrug, you measure the active metabolite. Those are not too difficult to deal with. When you get into these more complicated types of molecules, it gets a lot trickier, right? If you look at ADCs, what's the active part? Well, it's the payload, right? But the free payload that's released is actually very, very, very low. It scrapes the lower level of detection with most of the assays, right? And so what do? Well, we look at the ADC, the intact ADC, because what we find generally in those cases is that the disposition of the ADC is very similar to what you see for the free payload. So that's a reasonable surrogate to use, and we've had a lot of success using that moiety in exposure response analyses to understand how the drug behaves. Okay, so in the ideal sense, you want to measure that what you believe is the active moiety, the one that's killing the cancer. Okay, but sometimes it's not possible, and so we have to use another part of the molecule that tells us or reflects the behavior of the active portion of it, right? That's something that we rely on the sponsor to make a determination about. Okay, so the people developing the drug, they know the most about their molecule, so they have to tell us what the active moiety is, right? And we will get into discussion. They have questions about this. Coming back to the ADC example, people developed assays for the payload, for the ADC, and for the antibody, the total antibody. All right. Generally, what you see though is that the total antibody reflects the ADC, and the ADC reflects what you know about the free payload. All right. Do you need to keep measuring all those over time? I don't think so. We don't. So when people have asked, I've said, since everything is consistent and proportional, and we're using the ADC measurement, I don't see any value in continuing the total antibody per se. And if all things being equal, we don't need to continue monitoring the free payload per se once we understand, how it impacts toxicity.
If we could even measure it in the first place.
Right. And so it's probably fair to say that early in development, we may be doing a lot in terms of bioanalytical analysis, but as we progress over time, we can drop some of these other assays and just focus on the one type of or one part of the molecule to tell us about the clinical pharmacology behavior.
That was a point that Olin Stevens made in a CMC discussion that we had, that in the beginning, you may be measuring a whole bunch of attributes. Yep. You may not be doing it to, where there are specs and, set up already, but you're looking to try to figure out what is going to be the most, what is the critical quality attribute that you can measure consistently kind of approach.
Yep.
So would that be one of those, you're going to go on hold for your IND if you don't have an assay that's able to measure something that's reasonably correlated with the effective part of the drug?
No, we're not going to put you on hold for that. We put people on hold for safety issues, right? This is a characterization of things. It's important, but it's not a safety issue. We have had some rare cases where people didn't have their assays ready yet in their phase 1. Okay. And, in those individual circumstances, we advise they collect the samples and they store them until they have their assay finalized, and then they can do the kinetic characterization. We don't want to, that's not what we advocate, but sometimes these things happen and you just have to work with what you have.
Well, I guess Nan's probably going to talk about this.
Yeah.
But that doesn't seem like intelligent drug development if you don't even have an assay in place to measure, quote, the concentration of the effective moiety of the drug while you're doing your first in human trial to look to see. I mean, this is where I would point folks towards, I guess if you're just going for toxicity, but isn't the idea to try to look for hitting the target, hitting the biomarker, some measure of, how do you even know you got the drug? How do you know the protein binding of the drug? All those seems like critical measurements for good drug development.
And clearly, groups and companies that are very experienced in this area have a well-established paradigm for the sort of the groundwork that they have to have ready before they're going to start in a clinical trial, right? So they'll know about the P450 liabilities. They'll have their bioanalytical assay set up and ready to go. But for somebody who's starting out, like you said, this can be very daunting, and where do you look, right? So I can understand how that can get overlooked sometimes, because when you're thinking of how you're going to be dosing patients, that might seem like a secondary consideration to you. It's important. You do have to think about it, right? But it's understandable.
Is measuring the drug that's in the patient it doesn't seem like a secondary consideration. It seems like a kind of primary consider that the drug that a certain concentration of the drug was in the plasma, because I presumably have some animal studies to suggest that I want to reach a certain receptor saturation or AUC in the animal that would correlate to something in the human. Is that not
clearly we're preaching to the choir here, Jeff? I mean, we all know this, right? But in terms of somebody who's new and starting out, they may not appreciate that. And so I
guess why are we putting them on hold for that? Because that seems like
we are not putting them on hold.
I know, but should we?
Well, you got to remember, when it comes to putting people on hold, we do it for safety issues, right? Yeah. And so we're measuring the kinetics, and we want to use that information to understand how the drug behaves with respect to efficacy and safety, right? But it's not a safety issue directly.
But what you guys would tell anybody who's going to develop a drug who doesn't have an assay to measure the concentration of the effective moiety of their drug in the patient before they start putting drugs into patients, probably ill-gotten, ill-informed drug development. Is that fair to say?
I think the first in human study designs usually allow some uncertainties. With regard to drug properties, with regard to characterization, and many of these can be handled by revising the study protocol and smartly designed study protocols. In terms of characterization, for example, when you say we don't necessarily have a model method ready, and we do not usually before first in human study, we do not really have a very comprehensive understanding on the DDI potential. In those cases, we can sometimes adjust the starting dose and use some risk adjustment in the inclusion-exclusion criteria to make sure that we do get too much variabilities in terms of PK exposure and the safety profile. So, and I think there are things we can work on to ensure that patient safety is largely guaranteed with the study design. For the bioanalytical method, I think this will be pretty challenging, especially when it comes to the complex drug product. Sometimes the drug product you design do not really perform the way you want. Nanoparticles, they do not always serve the purpose for target delivery. So I think for the first in human studies, before testing in human, sometimes you cannot really tell. In those cases, I don't think it is always a big stop sign that you cannot move on. as long as we can use other measures, like by adjusting the dose with appropriate safety monitoring, some of the characterizations can be delayed or come up later after we gain some clinical experience.
I would completely agree. Drug-drug interactions, there's a lot of stuff that can be done at a later time point. It just seems kind of critical. I hate to perseverate on the point that if I don't know what the concentration, if I can't measure the concentration of the drug in the patient kind of at the get-go.
Sometimes it is the desirable effect for some of the lipsome product nanoparticles you wanted to go to the target site and do not have the free drug circulating. So it is, I think that sometimes it is a desirable effect, and we would expect difficulties in the direct measurement.
So, I would say we all agree that having these set up before you start, having your assay validated and ready to go before makes good sense, and we promote this regularly, and we see this most of the time. And I just want to be clear with the viewers that we're talking about really the exceptions here and how we can sort of deal with these exceptions when they occur. Right? So somebody didn't have the foresight to develop that assay. What are some of the things we can do moving forward with that? Okay, that's where we're coming from. But
we try to find flexibility to not be overly burdensome, to allow development plans to proceed.
We work with drug companies to work in a variety of different ways to accomplish the goals of drug development, right? It's not always nice and pretty the way we like envision it in our heads. Sometimes other things come up and we have to look at things in a different way. We might do the studies in a different way. So there's a lot of flexibility that goes into this. And this is, I think, really some of the most interesting parts of what we do here at the agency. And so we see these different things, and we work with companies to make sure that we're getting these questions answered that are critical to patient care.
Is there a forum to go back to the kind of complex drug products with multiple components, and what's the free drug nanoparticle with MER RNA in there and a targeting agent all wrapped up in something? Many times, for a pre-IND meeting, we'll just provide responses, see these various guidance documents. Is there a guidance document specifically on developing assays for your drug and when they're complex products, or is there a forum for interacting with you guys to be able for them to ask questions along these lines where it's not clear?
So the guidance that we have for validating our assay is the FDA M10 guidance. Okay, it's not specific to particular assays for particular drugs. Okay, it tells you what are the general principles and the general criteria you should follow to have an assay that's going to be successful for what we all need to do with this data. Okay. When it comes to like specific drugs and bioanalytical development aspects, what I advise and people have told many people is, when you have questions, submit them through the IND. Okay. So they can get the attention they need, so we can interact with you over those things. And, for all of these things, and not just the bioanalytical part, the agency is available. We have different meeting formats, right? So we meet with drug developers through the course of their development, and they ask us questions about the development of their products. So they can ask any questions about the clinical pharmacology data. We will interact with them either in a written format, or we could have like a teleconference or a face-to-face meeting. We don't do the.
Have you had any of these type D meetings, the new PDUFA 7, whatever, where they get to ask a limited number of questions that might be pharmacology based? Have you had those meetings?
We have had a few of those. Not that many. Nan, do you want to tell us about your experiences with some of those?
Yeah, we do have type D meetings. They can be a follow-up to any type of meetings, including pre-IND meeting may be very relevant to this discussion about first-in-human study design and initial IND submissions. Otherwise, to the other types, meetings, the sponsor are free to submit those follow-up requests. Sometimes the follow-up request for clarification can come in a Type D meeting format, and sometimes it can be not in a formal meeting format, but with depending on the contents. If it is very straightforward, simple question, single question, it may be submitted as a request for clarification.
Yeah, and another thing that sometimes occurs, this is a lot less formal though, is there are a number of scientific conferences and medical conferences that FDA personnel attend, AACR, ASCO, ASCPT, things like this. And we often interact with folks there, and they ask us a lot of questions at those things, and we can provide some generalized discussion at those things. But at those kinds of meetings, you have to keep in mind that those are very general discussions and they're not binding on anybody. And if it sort of points you in the right direction, the next best thing for you to do is to submit those questions and your plans and questions about your plans in your IND so they can be specifically addressed and become a matter of the record. So that's helpful for both the agency and drug development folks.
And also, I think for those complicated product within FDA, there's a lot of collaborations between the divisions, and there are working groups dedicated to those complex drug products. And a lot of times when there are meeting requests coming for specific drug products, those interest groups may be consulted to address specific questions related to characterization and things like that.
Are there actually things that they need to have? Because based on kind of our discussion so far, I don't think it doesn't sound like there'd be a reason that you guys would put anybody on hold for a first-in-human trial.
If there's a drug-drug interaction liability, that could lead to a hold if there are some issues with.
It sounded like we didn't necessarily look up front for drug-drug interactions. That wasn't necessarily a requirement.
So, like I said, most drug companies will do their screens ahead of time, so they understand what the liabilities are, and they'll share that information in their first-in-human protocol, and we'll look at the exclusion criteria, right? And if those are not being accounted for, those potential interactions are not being accounted for, we may say, hey, we're going to put you on clinical hold until you adjust your inclusion-exclusion criteria to make sure that we're not endangering anybody by a potential drug interaction.
So if you didn't do those, is it micronuclease assays? What are the in vitro or in vivo drug interaction studies? There's a guidance document on it, it sounds like.
There is, and it talks about the different in vitro types of assay systems that are, the isolated enzyme systems, and you can get an indication of whether your drug inhibits them or not.
And if you didn't do that, we wouldn't have that information and we wouldn't know. So all you could do is we're gonna enroll patients who are not receiving any other drugs. That would be a way to.
Yep. Nan, do you want to talk about that?
Yeah, I can talk about that. And as I said, at the time of first-in-human study protocol development, we do expect a lot of uncertainties in terms of the characterization. But those uncertainties needs to be addressed in the protocol design to ensure patient safety. For example, for the DDI information for the ADME properties, we do not know how much hepatic elimination may be involved. Then we may be requesting pretty restrictive enrollment criteria and providing pretty restrictive instructions with regard to concomitant medications. So there will be a balance based on available information and what you can actually enroll.
And it might be pretty hard to do an oncology drug trial with patients who are receiving no concomitant medications. So
that will be very,
that's true.
Yeah,
it
that
would
will
behoove you
really
to at
help.
least do some of these characterization studies up front, or you may not be able to accrue patients on the study because you're so limited on the
And who you can bring in, right?
yeah. As I like to say, CBER has some really funky drug products. Many times they're alive. We don't have to worry about those. But there are certainly interesting CDER drug products, those complex nanoparticles, bispecific, trispecific engagers. There are nucleotide drugs and MERS. And what do you guys see what are there for each particular drug class? Are there things you would want to point out to people that they would need to think about specifically, or is it read the guidance documents and we've addressed those there appropriately?
Well, one of the things you brought up were the T cell engages, right? Which is a relatively new modality that we're seeing in hematological oncologies, but also in some of the solid tumor areas. And some of the things that we're doing there are newer, right, with respect to these things. And one of the characteristics of these drugs is that they're prone to causing cytokine release syndrome and perhaps ICANS, which are can be very serious, even life-threatening syndromes in patients, right? So it's taken very seriously. And to deal with these, a lot of the times what we're doing is we're seeing step-up dosing before you get to the full clinical dose in order to prevent or reduce the onset of CRS. Okay. And so that's certainly some of the standard advice that we're talking about now with folks, is that they really have to look at a step-up dosing paradigm before they get to the full clinical dose. And we recommend people use QSP modeling. Okay.
So, QSP is
quantitative systems pharmacology modeling. Okay. And this sort of gets into a modality or a tool that we haven't talked about much, but that is the use of pharmacostatistical modeling and simulation based on those models, right? So we're using how is it? Computers and mathematical algorithms to understand to model the behavior of the data, and then we can use those models to ask what if questions, basically, and we can try different scenarios using these models. So you can look at different dosing steps, and so this has been very useful in the cases we've seen, because
is this like dose response, dose safety relationships, or is it dose biomarker, or is it all together?
It is all of that, right? So a lot of these models are developed in non-clinical models, and then they're taken forward into the human setting once we start to get some clinical data around the efficacy and the safety events that occur for these drugs. And so you update the mathematical models with this information, right? So that it behaves progressively better with that data. And then you can ask, like I said, what if questions. You can say, well, what if we dose it this way? What's going to happen to the efficacy, and what's going to happen to the safety? And using this has been very useful in helping to identify the dosing paradigms that you want to take into the clinic next to try to deal with CRS and get to a point where the drug is efficacious, right? So this is something that's novel, and we don't have any guidance on that right now because it's so new.
It also sounds like you need a lot of patients and a lot of data to inform the model to make it be reasonably predictive, which
it makes more sense for us to use all the tools we have so we can make progressively better choices, about the doses that we're picking to take forward in patients. Okay. And so there is, some added effort, and I guess clearly some added costs up early in the development. But I think from what we have seen, that has paid dividends, at least in terms of, the products that have been approved on the market for things like multiple myeloma and DLBCL and things like that. And the tools that have been evolving and emerging in this space have been very helpful to that, I would say.
And you call it, what was called QSP?
Yeah, QSP, quantitative systems pharmacology.
And how is that different than that MIDD?
Okay, so the MIDD program is the Model Informed Drug Development Program. Okay, and that is a broad banner that describes sort of the in silico types of approaches, the modeling and simulation approaches, where you ask what if kinds of questions. Okay, so it's a very broad banner that describes the overall program. QSP is one of the modeling modalities that people can use, right? People also do population PK and exposure response and PBPK, physiological based PK modeling. Okay, so there's different ways of there's different technical ways of using the data to answer different questions. And the MIDD is sort of the overarching descriptor of all of these different modalities.
Here's a little, it's not meant to be a curveball, but over my experience, I have seen many times where, there'll be various modeling, exposure response relationship stuff. And yet with an oncology drug, and I'm not sure why, you would think there'd be an exposure response, but many times there's not an exposure response. I'm assuming that's just that weren't enough patients and not enough data to inform that particular analysis. But what do you have to say to that when we don't even see exposure responses, and yet we're trying to integrate that into various decision making?
There are a variety of reasons. There are several reasons for what that is, and Nan's done a lot of this. So maybe Nan, you want to lead off and tell them what's are some of the limitations that we faced in this?
Yeah, there are many reasons why from a report something you do not see a ER relationship, which may be against what you expect. I think one of the many common reasons is that the sponsor may be conducting a formal ER analysis within a very small, very narrow dose and exposure range. Many times that is the dose level based on previous dose selection strategy, one single dose level, and you have a small, very narrow exposure range, and you cannot expand to see any trend, even if the trend is actually there. And sometimes because of this narrow range, it is also possible that in terms of the effect, it is already at a plateau. That's why it's not showing. If you are choosing too high dose, even if you choose 2 or 3 high dose, because they are all in the high range, you still do not see a relationship. And sometimes depending on the data, sometimes the sponsor may be using very early stage data, and where the disease, the patient comes from very different, diverse background, and there could be a lot of confounding factors that can constitute this data set. Many different disease background, patient characteristics may be masking some of the ER relationship within the data set. So I think those are the 2 major reasons, most commonly see reasons that you do not always find a positive ER in the report that is.
I guess you can't have a dose response relationship if you only have one dose, right? If you're swinging away, you think
difficult.
Yeah.
I think one of the things that has confounded that particular issue is, we used to pursue drug development by following the MTD paradigm, right? And so based on that paradigm, you want to give as much drug as you can to maximize the efficacy, with the proviso that we're going to deal with whatever the toxicities are. And I think what happens is a lot of the times you end up on the top of that exposure response curve, way far up, right? And so even when you see some changes in doses, you're so far up, you don't see where the curve starts to come down, right? So you're not in the best place to make a decision about where is that sweet spot between the safety and the efficacy.
And you're also much more likely to get off-target toxicity effects from your drug.
Yeah,
that
well,
you wouldn't need to.
if
You
you
wouldn't
keep
need
pushing
to.
the dose up, right, you're going to start seeing more adverse events of all descriptions. Yeah.
So one of the, as you well know, I'm sure both of you, and as I have experienced at various meetings, one of the biggest angsts out there in the drug development community is, Project Optimus. And I just, and it sounds like with some of the recent discussions in the Oncology Center of Excellence that people are starting to come around to the idea, that this shift from MTD to a more intelligent, informed, dose selection, and that we're not trying to be prescriptive. We're not trying to be bureaucratic or burdensome. We just want you to bring a different mindset to how you go about doing that. How would you guys suggest, even in a first in human trial, that they start to think about, dose optimization? Is it taking in vivo animal data and translating that to the humans, having assays ready to go to measure your drug concentration, following a biomarker, potential biopsies for a biologic if you're looking for, T cell recruitment into a cold tumor to make it a hot tumor? What would be your thoughts there?
So kind of all of that. So in the past, one of the things that struck me is that we do a lot of development in the non-clinical space, and then we sort of get to the clinical interface, right, and we kind of forget about that stuff
and go to MTD.
Right. And we've been trying to get drugs out quickly to help deal with these diseases that are afflicting patients, so you sort of can't blame people for that. But we're saying, in Project Optimus, you've got to use all of the information. That includes some of the preclinical information where you've developed information about target engagement, where you've looked at biomarkers in animals that you hope are reflective of the disease, and you try to help bring this forward into the clinical setting. And you use your biomarker data, you use your concentration data, you use your toxicity data in the patient, you use your activity or ORR information in the data, and you're using all of this in your calculus to help select the best doses. Good. Now, as part and parcel of that, like I said earlier, a lot of the MIDD modeling is started in the non-clinical space and it's expanded into the clinical space, and we should use these kinds of things. Okay. And I think one of the hard parts that people have sort of latched onto is, we used to do a 3+3 design to get to MTD. And a lot of the times we're encouraging folks to include more patients in their cohorts, okay? So that you can get a better estimate of what the toxicity profile is and what the activity profile is at particular doses, right? And this is sort of the hard part, because how exactly do you do that? There is no one answer, right? There are different ways of doing it. Some people will go up and pick up a dose and they go to the expansion phase and they'll look at 2 doses, right? That's one possibility. And some people will go to phase 3 and they'll randomize doses, or they'll do a run-in. So there's different ways of doing it. And I think people are often frustrated because they ask us, what it is exactly, and we don't have a standard answer that we can give you for your particular application. You have to look
or your
at
patient population, that size of that patient population.
Exactly. But it is very important that we spend time earlier on, looking at more patients, at least at some of those doses in the phase 1 trials, right, to try to get a better handle on what the activity and the toxicity is at the different doses, so you can make better choices in conjunction with all the information you have from the biomarker and target engagement and the modeling and those things.
I had one specific question, which, no drug is not toxic. Aspirin is toxic. But one of the questions was, we have a completely non-toxic drug, which, once again, are we going to have to do dose randomization, for dose Project Optimus, right? And I said, if we think we have the right dose selected based on whether it be in vitro, in vivo, AUC's engagement, but they don't, no one, they either don't have the resources or they don't want to do a randomized dose finding study, and I think it was a large patient population. It was like colorectal cancer. Obviously, we're trying not to be burdensome, and we were going to take, the totality of the data, in consideration. If it were truly a non-toxic drug, we probably wouldn't, and they look like they were having activity, early on. Would we require additional, dose finding studies for something like that? Or if it was minimal toxicity?
Every drug is different, right? And you have to look at the data that you have for that particular drug to make a decision now. And, we've had cases where people have come in, well, I'm not go down that road, but you would have to look at the specifics in this case, right? So you're going to do some dose escalation. You're going to get information on the toxicity and the activity, right? And if you had adverse event rates that were, total adverse event rates that were like, 5% or less, and they're showing great activity, you go like, okay.
Yeah. Okay.
That's pretty good.
We don't see a lot of that.
No, we really don't. But we do in each case have to make a decision about the risk benefit ratio, right? We have had cases where people came in and they only did one dose, and we're like, oh, you don't know that another dose isn't better, but we've been at a point where the activity was very good and the adverse events were okay in a general kind of sense, given the therapeutic area and the patient population, right? And so we have moved ahead, and I think in some cases we've had them do a little more research as they're going ahead into another dose by including another dose cohort and making a decision before they go through into the main part of the trial about which dose to pursue or whether they should be both.
Now, you guys have had innumerable conferences, large conferences on Project Optimus and optimal dose finding stuff, right? So hopefully you guys can provide me a list of those so I can put the links in the video that folks can go and educate themselves on, which once again, if you have to step up to the plate. And we provide lots of guidance out there and lots of educational seminars, and this would be one of those to to garner some more education on. But I think it's we're truly not trying to be prescriptive. We're not trying to be. We're just trying to have a different approach towards which as Rick Pastor points out, it may incur some additional costs up front, but depending on the patient population, the drug, the toxicity profile, the activity, that all of those are going to be taken into consideration into what would be most appropriate for additional dose finding studies. That's fair?
That's a fair summation of where we're at. Like we've discussed, there are different ways of doing this. But the message that we need people to understand is that we do need to do this. Okay, that investment early it will pay dividends in the longer run, most especially for the oncology patients.
Yeah. And it just truly it doesn't. I mean, I guess if you're still developing a cytotoxic drug, you might argue the point that we're just going to go to MTD because we're trying to kill rapidly dividing cells. But if that's your approach in drug development, I think you're likely not to be very successful. It's a targeted world today. Understand the biology of the disease and target specific biology, and you don't need to over target that and get off target toxicities. It's not.
Well, I wouldn't say there isn't a role for cytotoxics. It's not for me to make decisions about what mechanisms of action are of utility in the oncology space or not. I don't think, we sort of developed the MTD paradigm based on those cytotoxic modalities because it was often very difficult to distinguish between the toxicity profile and the efficacy profile, right? Excuse me. So it tended to make sense to use that there. I think you still have to look at that to see if there is that sweet spot between activity and toxicity, even with MTDs. It might be harder to see, it might be harder to find, and it may be
for targeted therapies.
But well, I'm talking about cytotoxics.
Oh,
right?
okay.
So you still have to look, okay, because you may be able to make some improvement there. Certainly in the targeted therapies, to me, the MTD doesn't really make any sense whatsoever.
Yeah.
We do need to understand those relationships and find the optimum between benefit and the adverse events.
And for AD, I meant classic cytotoxic. I think ADCs, I think directed cytotoxics, they're still cytotoxics, but I look at those as targeted cytotoxics.
Well, very definitely. You got a modality that targets an antigen on the cell, right?
Exactly. So before, I know that you guys have been gracious in extending your time here. If there were specific points you wanted to highlight for our particular audience, entrepreneurs and nascent drug developers, academicians trying to cure cancer, what would you try to get across to these folks?
Ask your questions early of us. Okay, that I think is very important general advice. We have different mechanisms for interacting with companies. Ask your questions early, right? Nan, I don't know if you would want to add anything. What advice would you give?
Yeah, actually, I think this is the most effective advice because all drug applications has their unique features. So I think contacting FDA early and utilize all the possible meeting pathways that you can be utilized will be most efficient to help with the development.
Yeah. Oh, so read the guidance documents, educate yourself, and then talk to FDA about your specific product and your specific development plan to get appropriate feedback.
Right. Look at those. Then tell us what your plans are and ask us what your questions are around your plans. We do this. This is what we do. Yeah. We're here to help development of novel oncologic agents.
Awesome. Well, guys, I so appreciate you taking the time to chat with us. And once again, hopefully this is just an informal discussion and that people can listen some insight from this. And I hope you guys have a great rest of the day.
Well, thanks, Jeff, for inviting us. This was a lot of fun. I enjoyed it.
Thanks, Nan. Have a good day.
Bye-bye.
Yep.